And welcome to the last day of class. Hard to believe, but this is our last day doing test 4 exam review and the final reviews, second hour. So yeah, today test 4 is actually due. So you have till midnight. It'll be posted around noon. We'll get that posted as soon as we can. You'll have plenty of time to take it. We'll be doing the review for it here this morning. Sorry. Quiz 10 is also due today. So we're kind of crammed here on time getting the last assignments in. And then in 2 days, the final is due. That'll also be at midnight. We'll get it posted earlier in the day, hopefully around noon there. There's a sample final already posted, and the key to the sample final is on there. It's the last page. Okay, there's 70 multiple choice questions, and the key for it is the last page there. There'll be a data sheet and a periodic table on your final. So you can look at that. If you had me for 351, you know what my data sheet looks like. It has some NMR data, some IR frequencies, NMR shifts. It has some atomic weights, electronegativities, things like that that you typically need there that I don't want you to memorize, but I expect you to know how to use. And also the periodic table has to be visible when you take an ACS final, so that'll be similar to that. Now our final is not an ACS final. We cannot do that online here, but the questions and the level of difficulty and the content, and of course it goes over 351 and 352, is similar to an ACS final, okay? Most of the questions are single concept, and you'll get that looking at the sample final. This sample final we put together years ago were actually old questions off old ACS exams, so that'll give you a feel for it there. But yeah, the data sheet periodic table will be on ours. Homework book challenge, you'll certify that on our final. It'll be a checkbox. If you end up being in one of those gap regions point-wise at the end, I may follow up with you with an email to see some of your problems that you've worked. So have those problems ready to go. I may have you scan in a couple and show them to me. That's how we'll handle that certification anyway. Questions on that, email me. We will be doing recitation and office hours tomorrow on Tuesday, so we're still available. We haven't abandoned you yet. Okay, so get some help if you need there. But yeah, your review and studying will help you out there, I think. All right, let's see where we left off last time was finishing up the citric acid cycle. And it's this beast here, the 2 carbons coming in from glucose. Remember, acetyl-CoA we formed from pyruvate. And that was a decarboxylation event that lost a molecule of CO2. So we need to lose 2 more here to account for all the carbons from glucose. And they're right here. We did this decarboxylation last time after we did the oxidation of isocitrate. The alpha ketoglutarate dehydrogenase is an alpha ketone carboxylic acid, which is similar to the pyruvate to acetyl-CoA mechanism. In fact, it uses thiamine and it uses NAD+. The mechanisms are the same, actually. You just have a little bit bigger molecule. So I'm not covering that one. But that gets us to succinyl-CoA, and that's a thioester at that point. Here's the 4-carbon dicarboxylic acid. The one's the thioester. Transfers the CoA first to inorganic phosphate, and then it transfers that phosphate anhydride, you can call, to GDP. GDP goes to GTP. That stands for guanosine diphosphate to guanosine triphosphate, and that transfers and eventually forms a molecule of ATP from there. Don't worry about any of that. You'll learn about more of that in biochem. We're just pointing out the key reactions 1 through 8, how they relate to what we've talked about. Here's succinate going to fumarate, and that's succinate dehydrogenase using FAD, taking off hydride and then deprotonating here. I'm not going to go through that. You'll learn about that later. You can actually do it. It's not too bad. You just grab a proton off there, flow the electrons in, then have hydride go into FAD. Similar to how we've been having the hydride go into NAD+. So it's not that much different there. And then fumarate to malate, that's just what? Hydration of an alkene. So there we're just protonating the alkene, bringing in water, and taking the proton off the hydronium ion. Of course, there's a stereocenter here. It is an enzyme that's chiral, fumarase, that does that. This last step I'll show on the board to remind you about the importance of these oxidation reactions. Malate to oxaloacetate. That's kind of a funny name, right? Here's the oxalic acid part, dicarboxylic acid, and here's the acetate part. And that accepts the aldol reaction from acetyl-CoA to form citrate. We already did one there. So yeah, let's do that and we'll call it good. The rest here, electron transport, I'm showing you how the reduced cofactors NADH, FADH2 react with coenzyme Q. You'll learn about that later. There's 4 complexes here in the matrix of the mitochondria that then eventually form water at the end. Okay, so that gets reduced. Electrons flow in, break the oxygen-oxygen bond, and protonate. Yeah, and you'll learn about the proton gradient that flows through ATP synthase that allows for inorganic phosphate to add to ADP to make ATP. This is where most of the ATP is made, in the matrix of the mitochondria. But yeah, we're not going through that. Stay tuned for 481. We're whetting your appetite for biochemistry there. Let's look at our last step here, step 8, and then we'll get into the sample exam for test 4 and start to review there. So let's come over to the sideboard here and do this last mechanism, step 8 of the citric acid cycle. You see it there. We've got malate here, which you could call hydroxysuccinate. Okay, this just has that common name, malic acid, hydroxy, and it has a stereocenter. Okay, it is S, I think there, yeah. And then the enzyme, malate dehydrogenase. So what does that mean? Well, we're dehydrogenate, losing 2 hydrogens. That's indeed an oxidation. And the cofactor for this enzyme is NAD+. NAD+ is a very common cofactor. It's bound and used by numerous enzymes to do oxidation reactions. Okay, and so here's our product, which is oxaloacetate. And so our challenge here mechanistically is, how do the electrons flow? Well, the enzyme has a base that can grab this proton. And when it grabs that proton, it liberates these electrons to come down. And then we've got to do something with these electrons and this hydrogen atom, right? Well, they come off and flow into this position on NAD+. This nitrogen does bear a formal +1 charge, right? It's bound to the adenosine diphosphate, and we're not drawing ours, so squiggle means, you know, something else attached there. But if those electrons flow in, we've got to flow this in and put that lone pair on nitrogen there. That gives us NADH, okay? So it puts that hydrogen at that position across from the nitrogen, the 4 position it's actually called. And what does this give? Yeah, it gives the ketone there. So it directly goes to these 2 products. There's nothing else to draw. The mechanism is relatively simple, but make sure you have the direction of the arrows correct there and how the base on the enzyme is working there. But yeah, if you can master that and show how you can use NADH in the opposite direction to do reductions, you'll be able to handle most of the mechanisms moving forward in biochemistry. Well, there's a lot of acyl transfers. There are other mechanisms like the aldol and the claisen that we've seen before. So those reactions will really help you out in the next semester there. So very good. I think we're done there for finishing the topics here in 28 and now metabolism. Okay, remember 28 was the lipid chapter, but these 2 things, glycolysis and citric acid cycle, are extra things here. So let's get the sample test up here and see what's on that. So we've got sample test 4, there it is. And let's click through that and see which ones you might have questions on. And, you know, I've got the key right here. And of course, the key is also posted for it. So you should be able to get help there and figure that out. So a lot of structural descriptive things. So D-glyceraldehyde, that's not D, that's L, right? That's on the other side. And this compound B is actually the same thing as A there. That's just what the Fisher projection means. You see you've got the S stereocenter there. This one is indeed the R stereocenter, so C is correct. D is not correct. That's not a stereocenter. We're tricking you there with the solid wedge there, but that's a ketone and an aldehyde. That's not glyceraldehyde. That is right there. The aldehyde and then the two alcohols, the one stereocenter there. Okay, number 2 here, the D series. Why? Because that stereocenter in the back there is R. And if you have the same orientation here, this bottom edge of the heterocycle is coming out at you, and this up position off the 5 carbon means that that is still the R stereocenter, right where the hand is there, okay? And that's why this is the D series. Okay, so C would be the correct thing there. Disaccharide from starch, which one is it? Not sucrose or spudose, maybe in Idaho that's true, D there. But maltose, you would say, is the disaccharide from starch, which is what? Which is alpha linked, 1,4 alpha linked, not beta. Cellobiose is beta linked from what? Cellulose, not starch. Given this Fisher projection, so altros, that's not a hexose we had you memorize, but can you go from the Fisher projection of any sugar and then go to the chair of it? And I always relate it to glucose. So glucose is right, left, right, right. So that means these two positions, 2 and 3, are epimeric with glucose, right? So here's up and down, you see. That would be those stereocenters right there and there for that and that, okay? And then the others are the same for glucose, right? So I think B is the correct answer there, okay? So up and down. What? Oh, alpha. Yeah, and the alpha position's down there. This one's beta. That's up there. Okay, so the alpha and beta in front of the name refers to the anomeric position. This one's beta altrose. This one is the alpha altrose you're asked about there. Okay, and it is D, less stereocenter right. So a lot of descriptive things here on chapter 4 with the biomolecules, okay, when we add in those ideas. Find the anomeric carbon here. And this is a 7-carbon sugar. We haven't covered those specifically, but you're given the structure and you're asked something specific about it, the anomeric carbon. Well, which one is that? It's the carbon with 2 oxygens on it, right? And it's in the cyclic pyranose form. So we've got what? This carbon right here, B, has an oxygen of the ring and that hydroxyl. This is actually a ketose, ketoheptose, because it is a ketone there and it's 7 carbons long, okay? Different than the other ones. All right. Let's see. It's not letting me advance there. Oh, there we go. Okay. Okay. IUPAC name for this one. Let's see, which one is that? Well, that's got the up position at the 4. So that is what? Galactose. And it is a pyranose. And is it the alpha one? Yeah. So I think it's C there. Yeah. Blood sugar antigens. We describe those, and this is just a lot of descriptive stuff. Yeah, they are tetra and trisaccharides, including fucose. That's that reduced form at the 6th position. It's a methyl group out there on some of those blood sugars. Will they mutarotate when they're cleaved from the protein? Yeah, because there'd be a hemiacetal at that point, right? And they're located on the surface of red blood cells. So I'd say all of the above there. Non-mutarotating, that would be one that's a full acetal, hemi. Ah, there's the acetal there, D. Covalent forces hold together proteins include all the following, except, yeah, amide bonds, disulfides, yep, cysteine. Carbon phosphorus bonds, no. That might be referring to DNA, RNA, except there it's phosphorus oxygen bonds. It's the diphosphate diesters. There's actually no phosphorus carbon bond. Those are in the Wittig reagents, but not here. Carbon bonds, yeah, those are in the individual amino acids, right? There's a lot of carbon bonds. So I'd say except the one that's not included there, I'd say is C. Okay, pH 7, neutral pH. If you have tryptophan, tyrosine, leucine, you've got all neutral side chains here. Tryptophan, remember, has the indole. Tyrosine has the phenol, but at pH 7, those, of course, are still neutral. If you get up, above pH 10, you can start to ionize tyrosine, but not tryptophan. Okay, that indole hydrogen will not come off. And certainly leucine's neutral there. And then you have the carboxylate terminus, which will be minus charge, and then the amino terminus on the left will be plus charge. And those cancel out. So I think you're 0 here. Yeah, at pH 7. But draw out the structure if you need to, and that'll help you remember, right? There's the 2 acidic side chains, aspartic acid, glutamic acid. The basic side chains are what? Lysine and arginine, and we should include histidine. If you're super acidic, you can protonate histidine also, the imidazolium. So be aware of that. General formula of a carbohydrate is what? Equal amounts of water per carbon. So you got to have the oxygen there. These aren't that. So I'd say that one there. H2O, same number of oxygen as carbon, so it's hydrated. That's what the name means, carbohydrate. Base pairing here. I told you didn't have to memorize A, T, G, and C. That helps. I never say, you don't have to memorize anything. A good memory is always helpful in any class. But how do you figure out, you're given cytosine here, C, and what? Hydrogen bond, donor, acceptor, acceptor. So what you need is a donor, donor, acceptor here. And that's right here, guanosine. Donor, donor, acceptor. So if you flip this around, you can see the hydrogen bonding pattern there between C and G. Okay, this is A. These are tautomers of that, and that's not correct. That would be donor, acceptor, donor. Okay, and actually Watson and Crick played around with the different enol keto forms of the base pairs. And they had to rely on an organic chemist at Cambridge to actually tell them about aromaticity and which enol form was correct. And then they lined up the hydrogen bonding complementary patterns. That led to their correct structure for DNA. Remember, I told you a little bit about the story there? Glucose, negative tolerance test, may or not produce. Therefore, it is a reducing sugar? No, it's 1,2 linked. Well, yeah, you need to know something about the structure of glucose and fructose. Fructose, the 2 positions anomeric. The 1 position of glucose is anomeric. So that looks correct there, but make sure you look at the other 2. It's 1,4 linked. No, that's not correct. Sucrose is an L sugar. Nope. Okay. Naturally occurring is a D sugar. So I think B is the most correct thing there. Okay. Let's see this reaction, NADH. We're giving you the cofactor and the starting material and product, and you're asked something about this. NADH acts as what? A hydride donor. Well, where's the hydride? Well, it's the hydrogen right here with its pair of electrons. The lone pair on nitrogen can come down and push off hydride, and then that can add conjugate addition to reduce there. And then look, there's the proton of acid that protonates what? The enolate for that second step. So that's fatty acid synthesis, and we're showing you enough of the reaction and asking you some details about the mechanism there. DCC, what does that do? Efficient amide formation under neutral conditions, what byproduct? Yeah, the urea byproduct. So the following is not formed upon DNA hydrolysis. You've got to remember what DNA is, right? It's a glycoside of the bases, the anomeric position on DNA. We're talking about deoxyribose, and you have the phosphate diesters there. You can hydrolyze all of those things, okay? And what will you get? You'll get the ribose, you'll get phosphoric acid, pyrimidine, pyridine bases will come off. Acetic acid? No, okay, that's not there. There's no acetate esters or amides. If there were those type of things, then yeah, you would get that, but not in DNA. It's the phosphodiester of ribose with the anomeric nitrogen bases. L-DOPA treat Parkinson's disease, cross blood-brain barrier. We're a neurotransmitter. What key reaction is used to produce this drug? Well, I showed you that asymmetric hydrogenation to make that chiral amino acid. That's how a lot of chiral amino acids are made commercially. You need an asymmetric catalyst to do that because they're chiral. The chirality comes from the catalyst. The others are not correct. Which compound is not a steroid? Well, that would be what? Insulin. Okay, so progesterone, aldosterone, um, cortisone. You can kind of hear the ketone names there. Those are all the common names for a lot of steroids, but insulin is a peptide. Single bile salts, I showed you that too. That comes from cholesterol. That's still a steroid. The steroid, we mean we have the 4 rings, right? The 2 or the 3 6-member rings and then the 1 5-member ring, and then different functionality on them. There is some functionality you need to know, have memorized, and that's the enone and the A ring for testosterone, and what the aromatic phenol in the A ring for which one? For estrogen or estradiol, right? The female hormone. Cholesterol just has a secondary alcohol in the A ring. Okay. Yeah, so know enough about that. Not all the biology of them, but a little bit of the basics of the structure. Insulin you should be able to rule out because we showed you that and we talked about insulin also, what it does there. Okay, yep, correct site for chymotrypsin. Chymotrypsin cleaves what? At the C side of the aromatic ones. So tryptophan, tyrosine, and phenylalanine. And so here's the C side of glutamic acid, not there. Here's C side of asparagine, N. Q, that's glutamine. The N and Q are a little bit tricky. Those are probably the hardest structures to remember there. But chymotrypsin, what we're talking about here, is proteolytic cleavage at the C side of the aromatic. So that would be correct, C there. Others are Watson and Crick. So there's a little bit on that story. They built the models and they had help from Rosalind Franklin's X-ray structure. They could see it was a double helix. Franklin and Wilkins hadn't figured that out yet. You know, it's kind of bad form on Watson and Crick's part for not sharing their insight with those coworkers. They should have now. That would be a serious ethical violation nowadays. The scientific ethics is much more codified now. Watson wrote a great book that you can read about there, the whole story. It's considered the greatest story of all time in science. It's the most important scientific paper ever published. Why? Because all of molecular biology, everything we know about heredity, and everything really you can say about biology, how it functions, how all the molecules in every living cell's made, is due to the insight from Watson and Crick. So you can't understate the importance of this. But yeah, they didn't have computer calculation back then, nor NMR. Okay, and of course Martha Stewart wasn't involved. Okay, yeah. So the write-on part, you know, we can make these as multiple choice now. Provide products here. We form the glycoside of isopropyl alcohol, and it's just that position, both alpha and beta. Full acetals there for that. Here's the oxidation. This is the Tollens test. So we'd form the carboxylic acid right here. Here we're forming the methyl ethers all over here. And you should form alpha and beta. Here we're showing right now beta, and this is the anomer of ketone. So at that 2 position is where the ketone is. But you can form the isomeric thing here where you have the methyl alcohol up and then this OH down. That would be the alpha anomer, and all of them get methyl ethers on them. Okay. Here we have the acyl triglyceride. We just hydrolyze that. That goes back to an earlier chapter. Carboxylic acid derivatives, saponification reaction. So you'll form glycerol, and then you'll form the sodium salts of all these fatty acids, 3 of them. Okay. DCC coupling will couple what? Alanine to phenylalanine, and we'll form ala-Phe. You can just say AF here if you want, but make sure you know what that means. Not FA. We're forming phenylalanine there. Oh, and it should be Bach protected and the benzyl ester still. So yeah, you need to draw out the structure there. for that one. If we had the deprotection conditions, if we hydrogenated with hydrogen and palladium and then treated with weak acid at the end, then we get the Bach off also. So remember your peptide coupling agents and your protecting groups here. Aldolase, here we have fructose 1,6-diphosphate. You need to go to the ring open form, show the ketone, and then do the retroaldol. And what are the two products? GAP and DAP. And there you can just use those acronyms. Make sure you know what GAP and DAP mean, or draw out GAP and DAP, doesn't matter. Here's the reaction we just did on the board, malate dehydrogenase. So we go to oxalic acid. And you can draw the name here or the structure. And you'll see that on test 4 and on the final too. Make sure you can go in between the names and the structures of different things. You use that idea nomenclature to help you keep them straight, I think. Draw this out. So you need to know what R, G, W, and P are. So what are they? Arginine, that has the big guanidinium thing. That's on the N terminus. Glycine, tryptophan with the indole. And what's P? Proline. That's the only cyclic. So you have those all drawn out there. At pH 7, which ones are ionized? Well, arginine is still protonated, so that will be plus charge on the side chain, plus charge on the terminal amino group, so that's +2, and then -1 on the C terminus, so overall charge should be what? Should be +1, okay? The right structure. So if we got time, we'll draw that out, but you should be able to draw out any of these simple peptides there. We can ask you a lot of things about that, changing the pH or, how many amide bonds, that kind of thing. I always start by just drawing out polyglycine and then adding in the side chains. If they're kinked downward, you have a dashed stereocenter there. Remember, it's always the S stereocenter, unless it's what? Cysteine. It's actually R, but it's still kinked downward. It's still an L amino acid. Provide electron pushing here. We're hydrolyzing the polymethyl ether here of what sugar is that? That is glucose. And it's alpha position here you're showing here. You're going to get both the alpha and the beta here. Once you protonate here, form the carbocation anomeric position, and only at the aromatic position. Why? Because these are ethers. These are stable. This, though, is an acetal. So once we protonate there, we get the extra oxygen resonance stabilized carbocation. So we only get the water going in there, so it's a hydroxyl, and draw both alpha and beta. Okay. Which one hydrolyzes faster? The alpha. That's because it's anti to the lone pair there on the ring oxygen. It can fill right into that sigma star directly. I only mentioned that. Typically, I don't test on that kind of thing, but yeah, that's a little follow-up question. I think we may have just given extra credit for that second part there. The main thing is the mechanism of protonating, form the carbocation, and then have water coming in on the top and the bottom for both the beta and the alpha anomers. That's the tricky part on that one, I think. Oh, and then we have a sequence analysis thing on an octapeptide here. And these are always a little tricky. You got to read through everything here. This would be harder to do multiple choice, but we could ask you about little parts about this, especially about, where things are being cleaved and what's going on here. You're told that these products that result from the trypsin digest, the tetrapeptide, tripeptide, you go on and further analyze this. This tetrapeptide gives MI and LF. So, methionine and isoleucine and leucine and phenylalanine there are part of the tetrapeptide. So let's see what's the rest of the data here. And phenylalanine is the site where trypsin will cleave, right? I'm sorry, no, that won't be where it will cleave. In fact, there's nothing. Trypsin cleaves what? Trypsin cleaves arginine and lysine. You don't see any of those here. Well, no, there's arginine right there. That's really the only one there. So no, where's K? There it is. Okay. So in the tripeptide, that's where you'd cleave there. So there's probably a K on the end here. And then we've got the Sanger reagent here on these digest products. So the Sanger thing, remember, is the fluorodinitrobenzene reagent that always tags what? The N terminus. So there's methionine on the end. So that's on the end there. So you probably have MILF for this tetrapeptide. You can figure that out from that data right there. So what's the tripeptide? Well, alanine is on the N terminus there because that's the Sanger test there. And then lysine K is probably on the right there because that's where you get the cleavage, right, with trypsin. And then arginine. Arginine's got to be what on the N terminus because that's probably where it came off, right? It wouldn't cleave if it was on the C terminus by itself. So when you link this thing up, you come up with R, A, G, K, M, I, L, F. So if you link these things up and go back and double check how trypsin cleaves, trypsin's cleaving right there on the end, N terminus end to give arginine. And it's cleaving right in the middle on the C side of lysine for that AGK thing. And then MILF is on the C side. Okay. Anyway, if you go through that and work, there are some homework problems like that. You got to make sure everything's internally consistent. It's good to propose a structure and then go back and double check everything. Okay, and read the questions carefully there if you're doing a sequence analysis. A lot of times it's trypsin, chymotrypsin, or partial hydrolysis of your peptide, and then the Sanger or the Edman sequencing will give you some extra information there. But yeah, I think it's RAGKMILF for the correct sequence there. It's probably all we need to do. Now this mechanism we did last time, and I'm showing you the whole thing here. So showing you the starting material and the product and the byproduct, right? So you go from the 6-carbon, isocitrate here down to the alpha ketoglutarate, given the cofactors, NAD+, and the name of the enzyme, dehydrogenase. So enough information there. So what do you do first here? Well, you oxidize right here to the alpha ketone, okay, with the NAD+. And then you have the beta carbonyl to this carboxylate. Then you can push the electrons off here, form CO2, and form what? The enolate there that you just then protonate. Put that proton back there at the alpha position of the ketone. We did this mechanism in the previous video. So if you want to check that out, you can. This is a more complex one because it has the 2 steps. First, the oxidation of the ketone, and then decarboxylation. But if we ask you at all about these metabolism questions, we'll give you a lot of information here. And you either supply a product or the conditions or say something about what might be occurring here. We're not having you draw out the whole thing. Well, it's multiple choice, right? So you're not drawing out anything in its entirety. You're just analyzing individual reactions. That's the key thing we want to get you to do there. We've got some of these worksheets here. These are harder problems, some of which we've had on old tests. Yeah, this is the one for the sugars. Let's see, did I skip over the other one? A little tricky on the advancing here. Okay. Oh, this is the one for chapter 26. Yeah, sorry, I got the chapters numbered wrong here. So yeah, this should be 26 for the sugars, sorry. So the amino acids, the key thing here is to analyze, cysteine, which is still an L amino acid, the amino groups to the left in the Fisher projection. And the other one that has stereocenters is isoleucine, has a secondary butyl group. And I told you didn't have to remember that. The naturally occurring one, I think, is S. And then threonine has a secondary alcohol in the side chain. Those are the ones there. But yeah, you can analyze that. Go back and look at the book, and you'll see how those are done. Here's an extra reaction that's not covered in your book. It's treating a methionine peptide. Here's the methyl sulfide of methionine right there with cyanogen bromide, which is this bromo cyanide reagent you're shown here. And you're given the product, and it cleaves here, and it forms this lactone here. So that's kind of a crazy mechanism. But what it involves is the sulfur here has lone pairs. It's nucleophilic enough to add the cyanide and kick off the bromide. That forms a sulfonium cation. That sulfonium cation is actually a leaving group. The oxygen here, the carbonyl on the C side, can actually come in and knock off the methyl sulfonium as a leaving group. Okay, and then you actually hydrolyze that. And if we got time, we can push the electrons here. But if you use this lone pair to kick off the sulfonium, you can see you can use water then to cleave right there, and that gives the lactone. Okay, that was the oxygen of the carbonyl right here, and then water came in and transferred the protons to give that product. So that's a tricky mechanism. I consider that a very hard problem. That'd be very hard to put it in multiple choice form on your test. So it's likely not to be in there, but it is good practice to go through that. And then the peptide synthesis, this is very similar to what was on quiz 9, where you had to do the peptide synthesis. So let's flag phenylalanine, leucine, alanine, and glycine. So, you start with the glycine, um, protected as an ester on the C side. Then you bring in the Bach protected alanine, and I think you know the rest of the drill. So you always have those repetitive 2 steps. The new amino acid going on the N-terminus, Bach protected. And then you have the Bach deprotection step, and then the addition of the next amino acid. So it's very sequential. The tricky thing about peptide synthesis is the side chain protections. Okay, we showed you some of those. You always bring those in protected form already. This is a worksheet for 28. This should be 28, sorry. So there's a couple harder problems and metabolism problems. We did a couple of these already here. Thiamine, this is the trickier mechanism. You got to form the thiamine anion and add in here. And yeah, you have the sulfur anion and the decarboxylation. So this is a very tricky mechanism. You can look through that handout that had that mechanism on it. Here's an aldolase. Instead of using a proton here to transfer, you're using a zinc cation. There's a number of cofactors that involve heavy metals in different enzymes. You'll learn about some of those later. But you give the same retroaldol products, DAP and GAP. If you're going in the forward direction, you'd want to form the enolate right here of DAP, and that nucleophile would add to the GAP aldehyde carbonyl, and then you'd snap it shut. There's your ketone of fructose. So if we ask you something more general there, we'll give you more information about the products or the conditions, and that's what you see there. Here we have a sesquiterpene. This is a 15 carbon going to cadenine, which is this 15 carbon thing here. And that's a little bit tricky to do. You start out the same way, allocation, and then you cyclize here from here over to here. That's a little bit tricky. You have to do a hydride shift and then bring this alkene down to form the bicyclic structure. And then you lose a proton right there to end the thing. But you can go through that. A lot of these terpene syntheses are quite tricky there. There's also a file that has the keys to all these worksheets on Learning Suite. But, practice them, work through them as you can, just on their own there. So let me go to the board here, I think. Is that all of them for that? Yeah. I'll go to the board and work a few more. We've got a couple minutes here for the first hour review. Let's see which ones we can do. Not going to do all of them. A lot of them we've, just recently covered. So let me point out some of the trickier ones here, maybe. Let's see, which one should we do? Yeah, okay, 15. So on the multiple choice. So when DNA is hydrolyzed, so what's the basic structure of DNA? Well, it's the phosphodiesters, right? And this is going up to the next position there, and then the oxygen here on the deoxyribose. And then the bases here. And the bases are nitrogen bases, so I'll just draw the basic structure there for DNA. Here's our 3' position, here's our 5' position. And that's the convention for keeping track of the phosphodiester backbone. And why is it DNA? Because you don't have the hydroxyl there, okay? So this is deoxyribose. 1, 2, 3, 4, 5. The structure would look like this. In the open form, you'd have the methylene right there, and then you'd have what? Right, and then this one at the bottom. So 1, 2, 3, 4, 5. Okay, so that is deoxyribose in the cyclic forms right there. You tip it on its side and you get this. Now the orientation means that this bottom edge is coming out at you, okay? You have a hydrogen there because that was an aldehyde, right? And it's an acetal, not with an oxygen, not an oxygen glycoside like the disugars and polysaccharides are, but it's the nitrogen glycoside. So all of these can be hydrolyzed, right, with water. And if you're protonating here, you can add water and begin to break these bonds and transfer protons out there. Oh, I should do the rest there. So, this goes down to another phosphate. I guess I should not have that there. Okay, yeah, there's the correct thing with the oxygen at the 3 position there. So yeah, you could hydrolyze there. Can you hydrolyze there? No, that's the 4 position, which means it's D sugar. It's up right there. Can you hydrolyze here? Well, yeah, we can protonate here and then kick off here, right? So if you hydrolyze, you'll get phosphoric acid or phosphate, you'll get deoxyribose, and you'll get the base. Okay, so 15. The correct thing was. I'm sorry. Which one is it? 16. This is problem 16. I had it wrong. Sorry. Acetic acid. That's not going to be formed from that. Okay. Let's look at a couple of the problems here. The problem 1 on the right-on part. Let's see. How about this one? So this sugar here, drawn kind of unconventionally as we've been looking at. No, it's kind of in the upper right there, and this is up there. So that is a D sugar. I'm not sure which one is this. This should be fructose, right? We've got the ketone at that position there, and then we have right. No, it is a different one. Right, left, right, left, right. Yeah, it is different there than fructose has that up right there. Yeah, so, but you're given the structure here, and you're told you have excess sodium hydride and excess methyl iodide. So every OH here becomes that, and then every OH can grab methyl iodide. But you should recognize initially you've got both the beta as it's drawn here, and you've got alpha because it starts out as the hemiacetal. So it's in equilibrium with its alpha one as soon as you put it in solution. Even if you had the purified beta to begin with, as soon as it hits solution here, it can what? It can mutarotate over to the alpha. It can actually change the stereocenter here once it's deprotonated because this can go in, form O minus there. That O minus snaps back because you want to get the 5-membered ring that's strain free there. So you can actually equilibrate through the O minus there. Sometimes students ask about that too. So what are the 2 products here? For full credit, you needed to show methyl ether there, methyl ether there, there. This one, which is the beta, like I said, and this one is also methylated. And then the other one would be this here, methyl ether, and then what? Methyl ether down here and up right there. So that would be considered what? The down relative to that position. So this is the methyl glycoside of what? Not beta, but alpha. Okay. So there's 2 for that one. So that's probably the hardest one, I think, on that one. Well, maybe the retro aldol there, but yeah, GAP and DAP, you can just say GAP or DAP on those or draw them out. You should remember those structures as trioses from there. And, GAP is what? Glyceraldehyde. And that's one I've told you should have memorized, okay? And then in glycolysis, it's phosphorylated. So you can see the phosphates on the beginning fructose to help you remember that. And then you know where they end up in the retroaldol products, I think. Okay, that's probably enough on that one. All right, also on the right on, how about number 3 there? We could do the structure of the peptide there too, I don't know. Now let's do this one mechanistically. These are probably the trickier ones. And these sugar ones always give you a chance to examine the structure and to talk about it. Oh, we've got that same methylated sugar. And let's see, which one is it? What sugar is that? Well, it's a hexose. And is it one we have you memorize? Yeah, because it's equatorial, 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 equatorial. So that's indeed glucose. It's the methylated version. And is it alpha or beta? Yeah, that's down, so that's indeed alpha. Okay. And you can isolate that one. This is a full acetal now. It's not mutating. It's not in equilibrium with the beta. Okay. It's just the alpha one. And you're asked here, what? You put it in water and acid. And what are the products? And which one's going to react faster? Label the starting metals. I have to draw the opposite anomer. Yeah, so you could draw the beta here, and then it asks you about which one would be faster in the hydrolysis. So what are the products, first of all? Are we taking off all the methyl ethers? No, just this one here. And the mechanism would involve protonation there, and then loss of methanol right there. And that will give us this oxygen stabilized cation. And you can draw the cation on carbon or on oxygen, doesn't matter, but that's why we ionize at that anomeric position. This cation is the more stable one, as opposed to that one, that one, that one. Those are all secondary and no resonance structures, right? And then this one's primary. Okay, so under these conditions, we're just ionizing at the anomeric position. To take off methyl ethers, or ethers in general, you need HI, hydroiodic acid, very concentrated, and heat. You can hydrolyze ethers, but much more extreme conditions, okay? But this is a special type of methyl ether, right? Because it's anomeric and it's part of an acetal. So here, how do we get the products? So here's water going on the top. And initially you'll have what? The hydronium ion and all the rest of the stuff on there. And then use water to take off that proton, and that will give you the beta one, and draw out the rest of the stuff there to be thorough. Beta there. How do we get alpha? Well, that would be where water can attack from the bottom here, and that will give you this one eventually as you get it off there. So yeah, alpha and beta. And what else do they ask you about? Oh, draw the opposite anomer here with the starting material. So the opposite anomer would have everything the same, except it'd be the methyl ether there, right? With all the other methyl ethers there. So that would be beta. That's the one that's not shown. So which one would react faster under the hydrolysis conditions, alpha or beta, and why? Well, it's the alpha one's faster because this lone pair is set up directly aligned, coplanar with the sigma star. And that's the anomeric effect, which I mentioned, okay? And that does hydrolyze faster. And that's also an indication of why cellulose is more stable than starch. Starch is all polyglucose alpha linked, and you can more easily protonate that and then bring the pair of electrons out and break that glycosidic linkage. In cellulose, these lone pairs right here and here are not coplanar with the sigma star, okay? So these are not in close proximity to fill in and knock off a leaving group if it's in the equatorial position or beta linked, okay? But if it's alpha linked, axial, you can get that anomeric effect. Now that's an advanced detail that probably won't be on your test, but you can appreciate that and maybe learn more about it in some other classes. Okay, let's see, other right-on ones. I think we're okay there. Probably end it right there. I talked about the worksheet ones. The last one has the mechanism of the isocitrate dehydrogenase we already talked about. The sugar worksheets have a couple other ones there. And again, the answer sheet is on there. So I think you're okay. Reviewing, work as many problems as you can in those chapters. By far, the most important chapters here for test 4 are what? 26, the amino acids, and 27, the sugars. 28, not so much. That's the lipid stuff. And then just a little bit on metabolism, okay? But these are the heavy chapters for the reactions and the structures of what you need to know for 4 there. So definitely, okay. Very good. Good luck on test 4. We'll review here for the final, here the second hour today. So let's take a 10-minute break. Very good.